Good afternoon, everyone. I'm going to start with a new topic, Hardy-Weinberg law, and the chapter is population genetics. So, mathematician Godfrey Hardy and physician Wilhelm Weinberg, they independently showed in 1908 that population gene frequency remained constant from generation to generation under a system of random union of gametes in fertilization, especially when the frequency of the heterozygotes are equal to twice this equation. P square plus Q square plus 2PQ is equal to 1. Now let's understand this one in detail. So we are talking about Hardy-Weinberg law, which states that the gene frequencies. So when I talk about gene frequencies, not only I, but the population genetics, when it talks about gene frequency, it is simultaneously talking about both allele frequency and genotype frequency. So let's have a basic background look at what these frequencies are. So allele, I have told you, I have explained it in detail in previous videos. So allele is the existence of binary form of alleles, binary form of genes, binary form of gene. It is known as allele. So ये जो situation है, it can take three variations. First, homozygous dominant, then heterozygous dominant, and then homozygous recessive. Homo dominant, hetero dominant, and recessive homozygous. So ये जो particular gene की तीन forms हैं, these are known as alleles. अब the topic in question, it is talking about frequency of each of them, कि particular individual या particular group के अंदर homozygous recessive की frequency क्या होगी. And some of you, if you want to know the basics of frequency in general, how many times it occurs, it's as simple as it can be. Frequency is the repetition of everything, repetition of a unit, repetition of a factor, repetition of an idea, repetition of a market principle. It is the repetition of everything. That's why I said how many times it occurs. So let's say we toss a coin in which the front side is head, opposite side is tail, and you know when you toss a coin, there are different frequency of how many times both head and tail appear. So let's say I flip the coin 10 times, out of which head appears 7 times and tail appeared 3 times. So the frequency of head, it is 7 out of 10. Yeah, simple. You can say the frequency of head is 7, whereas the frequency of tail is 3 in random flipping of order 10. वो आप मेंशन करना चाहे तो भी नहीं, but you got the idea. जितनी बार repetition होगा, that will be the frequency of that event. अब in the same topic, frequency of alleles will be how frequently, how many times this binary form, this allele, this shape, this structure, these number appear in a population or in a group of population. So let's say we can take another example here. I'm talking about a gene of fair complexion among negroids. So as you know, negroids are mostly of dark complexion. So ये समझना मुश्किल नहीं होगा. The frequency of fair complexion among negroids will be very less. So let's say this in the negroid population, the dominant gene will be of dark complexion. In that sense, to look for fair complexion in the negroid population and to cite the frequency, which will be very less. And let's say out of 100 people, 8 appear only once. So the frequency will be 1 out of 100 or 0.01. And in the same way, if we are talking about the mixed color or variations in dark color, so mostly will fall in this line, fall in the middle spectrum. So let's say 70 out of 100 people, they are of mixed complexion or variations in complexion. So the heterozygous dominant will have frequency of 0.07 and फिर जो लास्ट में है dominant वो बची हुई frequency उसकी होगी. So that's the equation, that's the idea. Frequency भी समझ में आ गया. And now let's again have a look at where we started. So according to Hardy-Weinberg, a population, the gene frequency of which will remain constant if there are no other factors, no other influencing factor which can be clubbed into evolutionary factors. So in the absence of all other evolutionary factors, the gene frequency of any population will remain constant, and that constants, it, it is described by this equation: p square plus q square plus 2pq is equal to 1. So आप लोगों को बेसिक तो पता ही है कि this is the formula of p plus q whole square, which when simplified will result in p square plus q square plus 2pq. Now अब ये समझते हैं कि ये एक्चुअल में है क्या. So allele, as I say it again, every offspring will inherit one copy from both mother and father. That's why there are two copies of every gene in a population. So gene will be same, well, but the copies, they will be two. Those copies, they are known as alleles. And we are talking about both allele frequency and gene frequency. Then let's have a broader application, broader explanation of Hardy-Weinberg law. So we know that there are two major factors that are worked in evolution: natural selection plus mutation, and both work in opposite direction. Natural selection, it try to weed out the best one, whereas mutation, it integrates variations in the population. And here the full feedback loop, it manifests in the form that if there are 4 individuals and natural selection over certain time, it will try to bring homogeneity in the total genetic makeup of the individual. वो कैसे? homogeneity, यूनिफॉर्मिटी लाने की कोशिश की जाती है बाय नेचुरल सिलेक्शन, क्योंकि इसमें फिर यही करेगा. इन द सिंपलेस्ट टर्म, 4 लोग हैं, A is not fit enough, so the gene of A will be eliminated, फिर बचे 3, then the competition will ensue, and again C is not better enough, फिर क्या होगा? सिर्फ B और D बचे, then again natural selection will act, and in the competition that will likely to be followed, इन दोनों में से B or D, let's say D survives. So here all the variations, they have been eliminated. This is the way of natural selection. So if variations are eliminated, the individual population will be devoid of the diversity that is inherent in the multitude of life forms. because it is the diversity that helps in the propagation of life. As I have said, uniformity is antithesis or the a condition which is not required for survival, because uniformity is like a death blow to life. If all the genes are uniform, then they are not likely to get uh, get beyond a certain length of time. Why? The answer is, when a population has uniform genetic composition, means सारे के सारे जीन जो हैं वो एक ही जैसे हैं, नोज भी एक जैसे, आईज भी एक जैसी, कोई वेरिएशन नहीं है, then the awkward factor, natural selection or nature, the bizarre and the unpredictable element here, bizarre and unpredictable element which is nature, it is not uniform, because nature जैसे आप जानते हैं, it's diverse, geographies are diverse, topographies are diverse, seasons and climates are diverse, everything is diverse. Then why the genetic makeup or genetic composition of life form should be uniform? It should not be. Diversity should be there, but it is it is an irony that the thing that is unpredictable and diverse in itself try to bring about uniformity in the life form. So we will talk about that later in some other chapter. अभी के लिए आप इतना समझ लीजिए that there are two factors, major factors. First, natural selection, which weeds out the best, or which brings about uniformity. But to thwart this attempt, to block this attempt of uniformity, nature also integrates mutations, and those mutations, they bring variations, or they bring the diversity, the much needed diversity. So यहाँ पे आप दो processes देख सकते हैं which are going simultaneously. First, natural selection, it is trying to bring about uniformity, and at the same time there are mutation which those forces, they are trying to bring in variations and diversity, and dono, they go hand by hand. Now again, to Hardy Weinberg law. So it says that the gene frequency of individual, कौन सा जीन कितनी बार किस population में कितने time आएगा, this gene frequency, it will remain constant, not only in individual generation but intergenerational, from generation to generation. This will remain constant if there is absence of other factors. There are no variations, no mutations, no genetic drift, no sexual selection, no natural selection, no gene flow, कुछ भी कोई भी फैक्टर नहीं है, तब पॉपुलेशन और उसकी जीन फ्रीक्वेंसी, इट विल रिमेन कांस्टेंट. सो इसका एक दूसरा जो इंटरप्रिटेशन है ये भी है that in the presence of other factors, gene frequency are likely to be influenced. वो कोई बहुत डिफिकल्ट मैटर नहीं है समझने के लिए. वी नो दैट अभी आपको बताया, जो नेचुरल सिलेक्शन, दिस ब्रिंग्स इन यूनिफॉर्मिटी, एंड म्यूटेशन, इट ब्रिंग्स इन डाइवर्सिटी. दोनों ऑपोजिट फोर्सेस हैं एंड दे वर्क इन टैंडम. ये कंटिन्यूस चलता रहेगा, और जब तक ये ऑपोजिट फोर्सेस हैं, म्यूटेशन जो है, इट्स अम्ब्रेला टर्म व्हिच इंक्लूड्स जेनेटिक ड्रिफ्ट, assortative mating, natural sexual selection, bottlenecks, inbreeding, outbreeding, ये सब मिला के an umbrella term mutation is formed. दोनों साथ में चलते हैं. और जो हार्डी वाइनबर्ग का दूसरा इंटरप्रिटेशन ये भी है that if other factors are present, then the genetic frequency will be affected, will be influenced in the proportion to the forces exerted by other factors. खैर, वो भी दूसरे चैप्टर में देखेंगे जब भी आएगा। फिलहाल आपके लिए इतना ही है। The gene frequency will remain constant from generation to generation. Why? Because of the absence of other factors. फिर ये जो कांस्टेंट है ये कैसे रहेगी? तो वो ये है कि लेट्स से दे आर टू alleles on a single locus. ये जेनेटिक पॉलीमॉर्फिज्म में वी हैव डिस्कस्ड इट। सिंगल लोकस क्या है? तो सिंगल लोकस पे दो अलील्स हैं, फर्स्ट ए एंड कैपिटल ए एंड स्मॉल ए, व्हिच मींस डोमिनेंट एंड रिसेसिव। अब ये है कि इनकी फ्रीक्वेंसी ऑफ dominant A will be P, and let's say frequency of recessive A will be Q। यानी किसी पॉपुलेशन में A जो है वो जीन फ्रीक्वेंसीज में P बार आता है और उसी पॉपुलेशन में रिसेसिव A जो है वो Q बार आता है। सो कुल मिला के हाइपोथेटिकल सिनेरियो इन विच जिस भी पॉपुलेशन की बात करें उसमें एक ही लोकस है, सिंगल लोकस इज देयर। सो सिंगल लोकस है और यही दो अलील ही हैं, दोनों अलील, डोमिनेंट वन ये और ये। अब ये है कि मैथमेटिकल फॉर्मुलेशन में वी सिंगल आउट द टोटल इज इक्वल टू वन। पहले बचपन में जब आप पढ़ते थे तो 100 रखते थे। नाउ इन मैथमेटिकल इक्वेशंस we bring about the whole system to one because we consider a system to be a circular one. So उस सिस्टम के जितने भी इंग्रेडिएंट्स हैं, जितने भी डिफरेंट एंटिटीज हैं, चाहे वो लैम्डा हो, डेल्टा हो, आईएनआर हो, जितने भी ऑब्जेक्ट्स हो, एस्ट्रिक हो, डिफरेंट ऑब्जेक्ट्स हो, लेकिन अल्टीमेटली व्हेन वी इनकैप्सुलेट इट, व्हेन वी एनवेलप इट इन अ सिंगल सिस्टम, सो अल्टीमेटली सारी डाइवर्सिटी जो है इट कैन बी काउंटेड एज वन। इन द सेम वे दूसरा आप एग्जांपल अगर आपको लेना है, whole world is counted as one, the earth is counted as one, whole Indian population is counted as one, American population is counted in one, the population of whole Maharashtra is counted as one। तो ये आप समझ सकते हैं कि वो इक्वेटिंग कैसे है। तो इसी इसी तरह से टोटल फ्रीक्वेंसी जो है यहाँ पे इज इट इज इक्वेटेड एज वन। नाउ लेट्स हैव अ लुक एट पनेट स्क्वायर। So Punnett square, it is used a diagrammatic representation which is used to derive the inheritance of genes in an offspring. So Punnett square इस तरह से, so this line is for male, uh, this row is for male, this one is for female। अब जीन की जिनकी हम बात कर रहे हैं उनके अलील दो ही हैं, डोमिनेंट ए, रिसेसिव ए, एंड दोनों की फ्रीक्वेंसी P एंड Q। अब ये मानते हैं स्पेशली इन हार्डी वाइनबर्ग लॉ कि पॉपुलेशन मेल एंड फीमेल के अंदर द रेशियो ऑफ डोमिनेंट एंड रिसेसिव बोथ आर इक्वल। दोनों के अंदर सिमिलर जेनेटिक कंपोजिशन होगा, यानी मेल के अंदर भी ये दोनों होंगे और फीमेल के अंदर भी ये दोनों होंगे, सिमिलर जेनेटिक कंपोजिशन। अब ये जो सिचुएशन है ये कैसे आती है? जो इनका ऑफस्प्रिंग है, क्योंकि मेल और फीमेल के गैमेट के कंपोजिशन जो जाइगोट बनेगा उसमें उसका जो जेनेटिक मेकअप है इट विल बी अ मल्टीप्लिकेशन ऑफ वन साइड ऑफ बोथ मेल एंड फीमेल। सो ए को ए से मल्टीप्लाई करें, सो द फर्स्ट ऑफस्प्रिंग विल हैव जेनेटिक कंपोजिशन ऑफ टू डोमिनेंट ए। फिर इसको इससे मल्टीप्लाई करेंगे ए, फिर इसको इससे मल्टीप्लाई करेंगे ए, और इसको इससे मल्टीप्लाई करेंगे ए। अगेन ए है, इसको आप ए स्क्वायर, इन दोनों को जोड़ के ए विल बिकम 2a a, and then the small one, both small, will square of small a. अब इनकी फ्रीक्वेंसी कंसिडर करते हैं. a की फ्रीक्वेंसी है p, सो दिस विल बी p स्क्वायर. यहाँ पे ये हो जाएगा 2pq और ये हो जाएगा q स्क्वायर. And since we knew that the total system equals to 1, so this is how we derive p square plus q square plus 2pq is equal to 1. That means total frequencies of population, यहाँ पे जो मेल एंड फीमेल की बात की जा रही है, मेल एंड फीमेल ऑफ होल पॉपुलेशन. और क्योंकि यहाँ पे वेरिएशंस है ही नहीं, पॉपुलेशन के अंदर जितने भी मेल एंड फीमेल है सबका जेनेटिक कंपोजिशन सिमिलर है, सबके अंदर एक डोमिनेंट ए है, एक रिसेसिव ए है. सो जो जितने भी ऑफस्प्रिंग होंगे वो इस रेशियो में होंगे, p square q square 2pq, and this frequency will be equated to 1. So that's how this Hardy-Weinberg law is mathematically represented. Now chapter पे वापस आते हैं. So ये आपको समझा दिया. अब what are the conditions of application of Hardy-Weinberg law? So in the population which is ideally large, first condition, with no overlapping generations, continuity in generation, sex is equally distributed, means वही genetic frequency जो आपको बताया, that both male and female, they have equal and similar genetic composition, and all parents are equally fertile. And when there are no changes in gene frequency due to mutation, gene flow, selection, genetic drift, and where mutation and selection rates are balanced, so there is no net change in gene frequencies. So इसका मतलब है इस total का, the first population should be large, then all parents need to be equally fertile, second condition, then genetic composition similar, and the last one, no other evolutionary factor, no mutation, no genetic drift, no gene flow, no selection. तो इस कारण से जो final population होगी, there will be no net change in gene frequency, it will be in the constant. Then proceeding on to the next paragraph, this theory is considered as the cornerstone of population genetics because it mathematically describes the behavior of genetic traits through time within a specific unit of the population. Actually, the population assumed under Heidi-Weinberg law is a unique population because, and not only unique but a hypothetical population. कि जो conditions हैं which have been mentioned here, those are not present, those are utopian and hypothetical. ऐसा नहीं होता है कि all parents are equally fertile, genetic composition भी similar हो. So we can say it's a unique population because it does not change genetically, it does not evolve, it's an ideal population or hypothetical one, which means that within it certain ideal condition must necessarily be fulfilled. So this ideal population is a mathematical abstraction because no real population ever fulfills all the necessary conditions. And those conditions are जो अभी यहाँ पे बताई, that population must be large, sexes means both male and female will be equally distributed, mating must be random, panmictic, all parents must be equally fertile, and then it must be free from four forces of evolution, which are mutation, natural selection, genetic drift, and gene flow. So these are the 5 conditions for application for abstraction of Hardy-Weinberg law. And UPSC perspective से आप ये चीज़ भी उसमें ज़रूर mention करें that the population considered here is an ideal population, hypothetical population, and the reason is because no real population will ever fulfill all the conditions. Then the Hardy-Weinberg law deals with the simplest genetic case, that of a single locus carrying only 2 alleles P and Q. ये आपको समझा दिया है कि जो organisms हैं in real world, they have होल जीनोम, और वो जीनोम कितना भी बड़ा हो सकता है, उसके अंदर बहुत सारे जीन्स होते हैं। एंड जेनेटिक पॉलीमॉर्फिज्म में भी हमने देखा है कि सिर्फ सिंगल लोकस नहीं है, बहुत सारे लोकस भी हो सकते हैं। बट ये केस जो है, इट्स अ सिंपलेस्ट जेनेटिक केस, इट कंसीडर्स अ सिंगल लोकस एंड दिस लोकस carries only two alleles, P and Q. Then the manner in which genetic stability is maintained under a two-allele system is best understood if the gene pool is visualized as divided into two component sexual units. इसका मतलब है जेनेटिक स्टेबिलिटी स्पेशली इन टू अलील सिस्टम, जब दो ही अलील हो, जो मैंने एग्जांपल लिए ए एंड ए के, दो ही अलील है इस टू, इन दिस टू अलील सिस्टम, द forethought means the precondition will be, ऐसा तभी possible होगा when whole population it is divided into two compartments, one unit that includes all the male gametes and the other unit which will be of female gametes. So the first unit, it will have the male gametes will have frequencies of allele P and Q, and female gametes OA है, उनकी frequency भी similar होगी. Then the relative proportions of P and Q are identical between two sex units. ये total जो यहाँ पे टफ लैंग्वेज है उसका सिंपल ये कहा है कि P and Q जो अलील्स हैं उनकी फ्रीक्वेंसी दोनों के अंदर सिमिलर होगी, जो आपको बताया सिमिलर जेनेटिक मेकअप. If all the male and female gametes mate randomly, the offspring will be distributed. So ये अब सिचुएशन ये कह रहा है that मेल एंड फीमेल के जो गैमेट्स हैं या उनका जो जेनेटिक कंपोजिशन है वो सिमिलर है. मैं इसको और सिंप्लीफाई कर लेता हूँ. लेट्स से ए एंड बी, सो ऑल मेल आर ए, ऑल फीमेल आर बी, एंड देयर जेनेटिक कंपोजिशन इज आल्सो सिमिलर. अब उनके बीच में मैं मेटिंग की बात करूं, दैट मेटिंग शुड बी रैंडम. सो how will that matter? How is that going to influence the genetic composition जब पूरी पॉपुलेशन के अंदर जेनेटिक मेकअप ही सिमिलर है, यानी ए और बी का क्रॉस सेक्शन ही होगा, तो जितने ही बच्चे होंगे वो सब ए बी ही होंगे. जब डाइवर्सिटी है ही नहीं, कोई सी डी ई एफ कोई अदर अल्फाबेट है ही नहीं, ओनली ए एंड बी है, तो रैंडम मेटिंग कराओ या उनका आर्टिफिशियल सिलेक्शन करवाओ, उनकी मैरिज करवाओ, इनकी कल्चरल सिग्निफिकेंस हो उसमें बीच में, एंडोगेमी हो, कोई भी अदर फैक्टर इंक्लूड करें, पॉपुलेशन के अंदर है ही दो टाइप, तो कितना भी उनमें वेरिएशन लाएं, कितने भी रेस्ट्रिक्शन रखें उनमें, कितने भी एंडोगेमस प्रिंसिपल यूज करें हम, प्रेफरेंशियल या प्रोबेशनरी मैरिज, जिस भी टाइप की मैरिज हम इंक्लूड करना चाहें, मीटिंग पैटर्न की बात कर रहे हैं कि मीटिंग चाहे रैंडम हो या ना हो, जब पॉपुलेशन में दो ही लोग हैं, तो दोनों की जेनेटिक मेकअप ही सिमिलर है, ए एंड बी ही है, तो बच्चे उनके ए बी ही होंगे, डिफरेंसेस होंगे ही नहीं. तो that's another caveat here. Then finally it says whether or not gene pool is initially in equilibrium, after one generation of random mating, genetic equilibrium at a single locus is established. And this again is not difficult to understand when we say all the other factors that are kept at constant, unchanging, or artificially uniform in their situation, same, the basic idea will be here, or basic objection will be here in the form that if at either at a single locus or either the whole genomic scale, if male are represented by A and females are represented by B, then the offspring will be AB. So this will be the first generation. Then variation will be included, but this first generation A and B leads to offspring AB. In second generation, क्योंकि वर्ल्ड में सारी जगह पे सारे मेल जो हैं वो ए है, सारी फीमेल रिप्रेजेंटेड बाय बी, तो सारे जितने भी चिल्ड्रन ऑल ओवर द वर्ल्ड ए बी, they will be born with this allele frequency, this gene frequency. फिर इनकी मेटिंग के बाद भी जब वही सेम पनेट स्क्वायर ए बी अगेन, फिर जो ऑफ स्प्रिंग हो गए उनके उनकी फ्रीक्वेंसी ए स्क्वायर, this one AB, this will be AB, and this will be B square. So second generation, the equilibrium will be reached that the total population will be one in the form that some children will be of A square, some children will be B square, and the remaining will be of frequency 2AB. So the population that is reached during such a genetic equilibrium will be in this form: a square plus b square plus 2ab is equal to 1, which is similar to this formula. So ये Punnett square आपको explain कर दिया था. So let us examine what would be expected under random mating in a simple and general case of autosomal locus with two allele A and A with frequency P and Q and corresponding genotypes AA, heterozygous A, homozygous A, heterozygous A, and recessive A with the corresponding frequency, this one. So again, this has expanded the same diagram. There is the same Punnett square, and same frequency estimation to the third generation. अगर आपको समझ में नहीं आया, A and B first generation, उनके children AB and AB, उनकी mating second generation. अब second generation में जो children हैं उनकी frequency A square, B square and 2AB. अब यही जो चिल्ड्रन है, यही ऑफस्प्रिंग है, इफ दे आर मेट अमंग देमसेल्फ, थर्ड जनरेशन से आगे का सोच रहे हैं, देन द सेम थिंग, ओनली इट विल ओनली लीड टू प्लीथोरा ऑफ नंबर, प्लीथोरा ऑफ बाइनोमियल, ट्राइनोमियल, पोलिनोमियल एक्सप्रेशन. जैसे ये जो चीज है, दिस इज नोन एज बाइनोमियल एक्सप्रेशन, ए एंड बी का होल स्क्वायर. This is a binomial expression which is represented in open form as a square plus b square plus 2ab. In the same way, when the numbers are more than 2, the characters are more than 2, then it will be known as polynomial expression. So अगर थर्ड जनरेशन के बाद फोर्थ जनरेशन में भी इनकी डिफरेंट फ्रीक्वेंसीज के साथ जो ये फ्रीक्वेंसीज हैं, दिस वन p स्क्वायर, pq एंड q स्क्वायर, जैसे भी आप इसको रिप्रेजेंट करना चाहें. अब सेम जब इनके बीच में मेटिंग होगी, मेल एंड फीमेल सिमिलर जो कंडीशंस हैं अंडर हार्डी वाइनबर्ग लॉ, ये करते हैं, अल्टीमेटली दिस सिनेरियो विल अपीयर, and the further simplification of which lead to the final conclusion that p square plus 2pq plus q square is equal to 1. So this table, it represents a formal demonstration or derivation that p square 2pq और q square is an equilibrium. So आप कितनी भी पॉपुलेशन ले, फोर्थ जनरेशन तक ले, फिफ्थ जनरेशन तक ले, ये पोलिनोमियल एक्सप्रेशन जो है ये बढ़ता रहेगा. यहाँ पे जो मीटिंग पैटर्न्स हैं पनेट स्क्वायर के अंदर, मेल एंड फीमेल में उनकी फ्रीक्वेंसी डिस्ट्रीब्यूशन बढ़ता जाएगा, but ultimately you will end up at the same equilibrium. So this is the beauty of Hardy-Weinberg law. And now let's have a look at the applications of Hardy-Weinberg law. So precisely, Hardy-Weinberg equilibrium, it postulates a set of conditions where no evolution occurs. So if all the conditions are satisfied, allele frequency will not change. And I have given you, you can say, a general but core answer that in nature there are two forces operating that govern the evolutionary dynamic. The first one is natural selection, the other one is mutation. Natural selection, it brings in uniformity, mutation, it brings in diversity, and इनके बीच का जो dual, it goes on, and this dual, which we know by the name evolution, is how life is controlled here. But Hardy-Weinberg law, it refutes, it negates the presence of both the disturbing or both the evolutionary factors. So इस condition में the situation is that there will be no evolution. सही है, वो आपने देखा है कि population will ultimately will set in an equilibrium, that when no evolution will take place and the permanent equilibrium will be maintained as long as this condition prevail. However, it is obvious that Hardy-Weinberg ideal population can never be found in the real sense. We know that जो conditions हैं they are not getting fulfilled, इसलिए जो ideal population है वो नहीं मिल पाएगी. But still उसके बाद भी Hardy-Weinberg law has some applications, had some need, क्योंकि if we consider this one as a standard which is not achievable, but then real populations, we can find their status by measuring the deviation from the standards. So एक particular population है which is deviating, real population deviating from the standard one by a margin of 0.2. So by measuring the population against a standard, we can know where the population in terms of genetic frequency is moving. So first application, this formula, it provides a standard against which genetic change in a population may be measured and predicted. ये first application है. Second application may be applied to large population to provide an estimate of gene frequency at a single point in time. Yes, the second application is also a better one, because in large populations where there are variations, diversity in various factors are getting in that we can measure or the gene frequency of a single locus. वो चाहे किसका भी, आप skin का ले लें, especially of those traits that are controlled by a single gene. So जितने भी traits हैं, let's take the example of eye color. Well, eye is a gene which is influenced by multiple other loci, multiple other stations. So this cannot be considered in, but there are many genes, many character traits which are controlled, which have a single locus. And this formula will have certain applicability in measuring the genetic frequency of those locus. Then population genetics is the study of allele frequency in a population organism of same species in same geographical area. ये एक बॉक्स है. The gene in population comprises its gene pool. कुछ terminology है जो अगर आप मिस हो गई है तो यहाँ पे understanding है. Microevolution reflects change in allele frequency in population. So इसको उल्टा करके देखें तो allele frequency, the change in allele frequency in a population is known as microevolution. Then it is not occurring if allele frequency stay constant over generation, which is Hardy-Weinberg equilibrium. और इसका simplification ये है that these 5 factors, non-random mating, gene flow, genetic drift, mutation, and natural selection, they bring in change in allele frequencies. And that change in allele frequency, it is known as microevolution. खैर ये अगर simple terms कहीं miss हो गई है तो अभी देख लीजिए. Now next one is deviations from Hardy-Weinberg law or the factor affecting gene frequency. So the ideal population of Hardy-Weinberg law is a large one and population which mate randomly without any mutations. However, in genetic disorders, several factors can disturb the Hardy-Weinberg equilibrium genetic disorder by influencing either the distribution of gene in the population or altering the gene frequency. अब इसका मतलब समझते हैं. Let's suppose in a population there are genetic disorders that are spread in various frequency. Genetic disorders, they get in via two mechanisms. The first one is mutation and the second one is expression of recessive traits. recessive traits. अब जैसे इस हार्डी वाइनबर्ग में भी देखा, the frequency of dominant homozygous is a square, then the frequency of heterozygous dominant is AA or PQ, then the frequency of recessive is small a square. And recessive are those genetic variations which are either non-functional or maybe lethal sometimes. So some such genetic disorders which prove lethal, they are known as genetic disorders. Let's take example of Turner syndrome. Turner's syndrome. So this is a syndrome in which the genetic makeup of the offspring is expressed in this form: 44 plus X followed by a void. So usually मेल के केस में आपने देखा होगा ये एक्सप्रेशन इस तरह से होता है फॉर मेल्स, एंड फीमेल के लिए 44 प्लस XX. तो एक X क्रोमोसोम तो होता ही है, बट टर्नर के केस में द अदर क्रोमोसोम इज मिसिंग. और अब ये है कि इसके कारण ये जेनेटिक डिसऑर्डर्स हो गया. इसके कारण द जीन फ्रीक्वेंसी ऑफ the whole population will be disturbed. First, usually it happens that the individuals suffering from Turner syndrome, they are either infertile or they do not complete the age much beyond their puberty. So all in all, they are either sterile or do not live up to much age. But if their genes are transmitted, their offspring, they will survive in healthy way. But as I have said, nature, it try to integrate the heterozygotes, which means nature will prefer the presence of one dominant and one recessive instead of both the extreme, instead of both homozygous dominant and homozygous recessive. So in that case, the offspring of the individual suffering from genetic disorder, even if they are healthy, they also possess the recessive gene. So उस कारण से जो genetic frequency है whole population की, it will also get disturbed. And second, if those genetic disorders they are caused by mutations, so mutation जो है वो ultimately genetic frequency को disturb करेगा, because ये mutation it comes from outside, because it is not similar to the working of natural selection. जो mutation होगा, just as, let's say from exposure to radioactive substances, जैसे हो गया कि यूरेनियम हो गया, कहीं पे लीकेजेस फ्रॉम न्यूक्लियर पावर प्लांट हो गया, या किसी भी तरह का रेडियो एक्टिव एक्सपोजर है, तो इट विल डेफिनेटली लीड टू सम म्यूटेशंस. लेट्स से स्मोकिंग, स्मोकिंग हो गया, या फिर जितने भी ड्रग यूज हैं, दे आल्सो ब्रिंग्स इन म्यूटेशंस. अब ये जो म्यूटेशंस है, दे आर ब्रॉट फ्रॉम आउटसाइड. सो फर्स्ट द लोकस एंड द पैटर्न इट विल नॉट बी डिटरमाइंड. अगर कोई फ्रीक्वेंसी के अंदर ही चल रहा है, जेनेटिक फ्रीक्वेंसी डाउन द लाइन, एंड इफ देयर आर सम मिसमैचेस और रीशफलिंग, वो है तो इट विल अल्टीमेटली फाइंड इटसेल्फ इन द बैलेंस. लेकिन अगर इफ समथिंग इज ब्रॉट फ्रॉम आउटसाइड, दैट थिंग इज टोटली न्यू अरीना, न्यू ब्लैक होल, फिर उसकी अपनी फ्रीक्वेंसी होगी एंड उसके अपने ही इन्फ्लुएंसेस होंगे. खैर, the factors which disturb Hardy-Weinberg law are non-random mating, mutation, selection, small population size, and gene flow. Let's have a look at non-random mating. So random mating or pan mixes is refers to the selection of partner regardless of the partner's genotype. So this one, it happens in an ideal condition. Usually, the mating pattern in the world we live is a non-random type. Non-random means we usually select the one who we are going to have children with, एंड उसी के लिए जितने भी मैरिज रेस्ट्रिक्शंस होते हैं, एंडोगेमी होती है, ये है जीन पूल होता है, कल्चरल रेस्ट्रिक्शंस होते हैं, इसकी वजह से वी यूजुअली एम्प्लॉय नॉन रैंडम मेटिंग, क्योंकि सिर्फ बच्चे ही पैदा नहीं करने हैं, उस पर्टिकुलर इंडिविजुअल के साथ रहना भी है, वी नीड टू मैनेज द फैमिली एंड एंश्योर इट्स सर्वाइवल। सो उसके लिए बहुत सारे फैक्टर्स होते हैं, और जनरली द एरिया ऑफ इन्फ्लुएंस और एरिया ऑफ एक्सेस ऑफ मेटिंग पैटर्न इट इज लिमिटेड फॉर एन इंडिविजुअल। अब इंडियन है, वो नाइजीरिया में अपनी बहू थोड़ी ढूंढेगा। In the same way, this area of access is a smaller one. So in short, we can say that here the population size is small. The population size in which we are looking for mates, it is small. And this thing is also applicable to all the individual, all the people who are living in certain area. So ये area है, let's say you are from Sikkim. अब सिक्किम यू कैन कंसिडर इट एज अ क्लोज एंटिटी। अब सिक्किम के लोग हैं, भले ही डिफरेंट डिस्ट्रिक्ट्स के हैं, डिफरेंट प्लेसेस के हैं, दे आर लुकिंग फॉर देयर मीटिंग पार्टनर्स, देयर मैरिज पार्टनर्स विद इन सिक्किम इटसेल्फ। सो देयर एरिया ऑफ एक्सेस और देयर पॉपुलेशन साइज इट इज स्मॉल। और यही चीज़ जो है, यही धीरे धीरे रोटेट होती रहेगी। पेरेंट्स जो हैं, ग्रैंड पेरेंट्स हैं, उन्होंने भी सिक्किम के अंदर ही अपने पार्टनर्स जो हैं ढूंढे। देन योर पेरेंट्स आल्सो फाउंड देयर पार्टनर्स इन द सेम एरिया, देन यू आर आल्सो गोइंग टू फाइंड योर मीटिंग पार्टनर इन द सेम एरिया। सो इसकी वजह से क्या होता है, जब पॉपुलेशन एरिया स्मॉल है, पॉपुलेशन साइज स्मॉल है, this event of non-random mating, it can lead to increase in the frequency of affected homozygotes, especially the recessive ones. तो ये आपने देख, यहाँ पे समझ में आ गया कि जो जेनेटिक पॉपुलेशन है, उस जेनेटिक कंपोजिशन है ऑफ इंडिविजुअल ऑफ दैट पर्टिकुलर एरिया, इट इज नॉट चेंजिंग। सो पॉपुलेशन जो है वो तो चेंज होती जा रही है विद टाइम। सम पीपल आर डाइंग, सम न्यू आर कमिंग अप, लेकिन जो दूसरे जितने भी फैक्टर्स हैं, जीन फ्लो, माइग्रेशन, वो कोई नहीं है। और आप मीटिंग जो है वो आप वही उसी एरिया से, यू आर ड्राइंग योर मीटिंग पैटर्न, मीटिंग पार्टनर्स फ्रॉम द सेम एरिया। सो अल्टीमेटली दिस बिकम्स अ क्लासिक केस ऑफ इनब्रीडिंग, क्योंकि जो इन्फ्यूजन है ऑफ न्यू जीन्स, इट इज नॉट हैपनिंग। बाहर से, दूसरे एरिया से genetic diversity is not being brought in. So this lead to expression of a situation in which homozygotes they increase in size. और अभी आपको बताया था, nature it prefers heterozygotes more than homozygotes. So in small area without any infusion of new genes, the homozygous they will increase in frequency. और अगर वो फ्रीक्वेंसी में इंक्रीज होता है, तो इसका मतलब है the offspring वो इस पैटर्न को they will also increase the same already increased frequencies. So ये एक इशू है नॉन रैंडम मेटिंग का। फिर it is written here the mechanism by which the frequency of affected homozygotes or recessive can be increased. So these two mechanisms are assortative mating and consanguinity. So assortative mating is the tendency for human beings to choose partner who share characteristics such as height, intelligence, racial origin for marriage. So ये आपको बताया कि marriage restrictions हैं आपके, all of those factors, all of those features and attributes that you artificially consider while selecting your mate, they are known by this umbrella term assortative mating. और वो है ही है, जैसे सिक्किम का ही बात करते हैं या इवन बंगाल की बात करते हैं, वी आर टॉकिंग अबाउट पीपल ऑफ वेस्ट बंगाल. So because of the um, cultural integrity, they are most likely to find a Bengali than people from other culture. ये तो एक ओपन सीक्रेट है, ये हर कोई जानते हैं कि एक मराठी जो है will look for a मराठी bride, मराठी groom. एक तमिल है वो भी तमिलियन ही ढूंढेगा, कश्मीरी कश्मीरी ढूंढेगा, सिक्किम का ढूंढेगा. So this affinity, marriage restriction, or most of which are social and cultural in origin, they will try to restrict the choices in gene pool available. And the second factor which may increase the frequency of homozygote is consanguinity. It is a term which used to describe marriage between blood relatives who have at least one common ancestor no more remote than the great great grandparent. So again, consanguinity mean marriage between mating between blood relatives, and this practice, this marriage practice, is prevalent in many societies when people marry their own blood relatives. So इस केस में भी what we are seeing is not a cultural practice but a shrinking of gene pool, a shrinkage of gene pool, because you are not looking wider, you are not looking for mates, um, you are not accessing the whole gene pool available at your doorstep. Ideally अगर देखें तो mating pattern में you have the whole world the whole world to mate yourself, but there are challenges, especially the social and cultural one. Linguistic challenges are there, adjustment are there, temperament, compatibility, बहुत सारे issues होते हैं. Okay, but at least we should have a horizon of at least 100 kilometer, a diameter of gene pool. It should spread at least 100 kilometers so that your offspring will not inherit the recessive, will not inherit the homozygous, because homozygous, they do not help in survival much. And the whole population genetics, the whole equation of life, both in philosophical and psychological terms, and even in general term, life is about living. What life there will be if you or your children who have inherited such recessive, such homozygous, suffer your whole life just because your culture did not permit you to find your mate outside that pool. Then consanguinity, it is a different affair. So I'm talking about looking for partners in at least in an area which span 100 kilometers all around. But this practice, it is about looking for mates in the immediate proximity, which is among the blood relatives. So इसमें तो blood relative में consanguinity में तो homozygote का inheritance, it really increases in frequency. So this is another case. Then let's talk about mutation. The validity of Hardy-Weinberg principle is based on assumption that no new mutation occur. So if a particular locus shows a high mutation rate, then there will be steady increase in proportion of mutant allele in the population. So again, this law will not achieve equilibrium. और ये आपको समझाया कि म्यूटेशन जब भी होता है, it brings in diversity, and that diversity is brought from outside. So this mutation will increase the frequency of mutant allele, which is a new allele other than what is already present in the population. So ऐसे केस में जो when we consider two alleles P and Q, now there is another R. So इस कारण से the equilibrium will get disturbed, will not be achievable. Then selection. So in ideal population, there will be no selection for or against any particular genotype. But in reality, for deleterious characteristic, there are likely to be negative selection. Deleterious means lethal genes. जैसे अभी आपको बताया टर्नर सिंड्रोम 44 प्लस एक्स एंड वाई. अब इस सिचुएशन में the population, it is not going to carry this mutation or carry this disorder. So what happens? Nature generally eliminates such genetic frequencies. So this is what is meant by negative selection. Then nature also works in another way, that such genetic disorders, such mutations will have reduced reproductive fitness. So as I've said, most of the genetic disorder, the individuals suffer from it. They do not reach the age of puberty, and even if they reach, most of them are sterile, which is the natural factor or natural selection at work to inhibit such individual from transmitting their defective genes. Then in the absence of new mutation, this reduction in fitness will lead to gradual reduction in frequency of mutant gene and will cause disturbance of Hardy-Weinberg equilibrium. और इसका मतलब वही है, same, that natural selection, it tries to bring in uniformity, because ultimately it will try to eliminate the less fitter one. So out of 5 it will keep selecting either positively or negatively 3 out of 5. Then out of those 3 it will try to select 2, and again the best one. This natural selection, it tries to bring in uniformity of genetic frequency, and that uniformity in genetic frequency is also not good for life. वो आपको समझा दिया है. So this uniformity, it is broken down by presence of mutation. So here the much needed diversity is being brought in. So if mutations are not there, in the absence of new mutation, the fitness, the reproductive fitness of the population, it will reduce, and the frequency, genetic frequency, will also reduce. Then selection can also act in opposite direction by increasing fitness, which is positive natural selection. So for some autosomal recessive disorder, there is evidence that heterozygotes show a slight increase in biological fitness as compared with unaffected homozygotes. This is known as heterozygous advantage. वो आपको बता दिया. So best example, sickle cell disease. इससे पिछले वाले वीडियो में, in which homozygotes have severe anemia, whereas the heterozygotes are relatively immune to infection with Plasmodium falciparum malaria. ये आपको उसमें बताया था that usually the erythrocyte, red blood cell, they are in circular in shape, but due to sickle cell anemia, sickle cell disorder, red blood cell का जो शेप है वो सिकल के फॉर्म में हो जाता है. So here in that case there are two variations, two allele. First one is for spherical shape and the other one for sickle shape. अब जो वेरिएशंस होते हैं, फर्स्ट होमोजाइगस डोमिनेंट, कि दोनों जो हैं स्पेरिकल शेप में है, जो मोस्टली अदर पॉपुलेशंस में होते हैं. देन द हेटेरोजाइगस डोमिनेंट, कि dominant and recessive both are present, but because of dominance of this gene, it will be expressed, and the final one will be the recessive one, in which both the allele, they are of recessive nature, homozygous recessive. अब मलेरिया के केस में, जब जिन भी एरिया में मलेरिया एंडेमिक है, वहां पे देयर आर टू काइंड्स ऑफ पॉपुलेशन ऑफ पीपल विद two different alleles. The first heterozygous dominant and the second one is homozygous recessive. अब ये वाला जो है इसमें जिसके अंदर दोनों ही जो alleles हैं both are for sickle shape. In that case, the person will suffer from hemolysis, anemia, and ultimately this condition can be lethal, dangerous. But the heterozygous, in which there is a presence of one dominant spherical shape along with the recessive sickle shape, in such a case, the dominant will express itself and the individual will survive without much hiccups. So this is a classic case of heterozygote advantage. Then, therefore, in those communities, there will be a tendency for the proportion of heterozygotes to increase relative to proportion of normal and affected homozygotes. ये भी is not difficult to understand कि वो सब population जिसमें दोनों जो है they belong to homozygous recessive. So ये वाली population versus heterozygous dominant. अब इस population में 100 लोग जो हैं वो heterozygotes हैं and there are 10,000 who are homozygous. So who is going to survive and who is going to spread gene for much time? And the answer is not difficult. This is a deleterious gene. Here negative selection will take place and these deleterious allele they will be eliminated from the gene pool, and these 10,000 people they are not likely to survive much, but they can survive if they mate with one with the dominant one and the resultant they will have a heterozygote composition, then the offspring can survive. Other than that, ये जो certain individual which have derived both the homozygote recessive, they are not likely to take up much time. But the heterozygote one, even if they are 100, they will live up to good age in a healthy way and they are likely to pass their gene, the heterozygous gene, in the future with greater frequency. So जो आने वाली generation है उनके अंदर heterozygotes will be selected, will be at advantageous position, and these deleterious gene they will be eliminated. Then there is also the opposite situation, that is selection against heterozygotes. वो कब होता है? The classic case is maternal fetal incompatibility, especially this disorder is known as erythroblastosis fetalis, which is observed in allele R, Rh blood group, and also for other group gene Rh ABO incompatibility. अब ये वाला जो है, ये वाला आप ऐसे समझिए, if the parents, mother and father, if mother she has Rh positive and father has Rh negative, which is a heterozygotic condition, but this condition will lead to destruction of embryo. So this disorder, destruction of embryo, it is known as erythroblastosis fetalis, which means the fetal cells of the embryo they will blast in fetus. So यहाँ पे जो केस हैं ये heterozygous एडवांटेज से उल्टा है, इसको कह सकते हैं होमोजाइगोट एडवांटेज, कि यहाँ पे अगर दोनों मदर RH पॉजिटिव है एंड फादर भी RH पॉजिटिव है, then the fetus will be unaffected. So उसमें फिर ये disorder will not work against. This situation, this is an example of homozygote advantage, a different case, other than usually or in the greater extent, heterozygotes are a definite advantage. So now the box mutation, they alters genotype frequency, by introducing new alleles. Heterozygotes and new mutations maintain the frequency of deleterious alleles in population. ये सारा समझा दिया है, just I guess it's a revision. Then different alleles are more likely to confer survival advantage in different environments. ये यही principle है of diversity, of natural selection, of heterozygosity or heterozygote advantage. जितनी diversity होगी, they confer a survival advantage in different population, different environment. Then in balanced polymorphism, a disease-causing allele persists because heterozygotes resist a certain infection, especially in sickle cell anemia. Balanced polymorphism भी देखा. The gene flow, it alters genotype frequency by adding and removing alleles from population. Declines are gradual change in the allele frequency between neighboring populations. Geographical barriers and language differences often create great differences in allele frequency, which is also a geographical and a cultural barrier. Then genetic frequency occur when a subset of population has a different allele frequency than the parent population or the larger population. And the founder effect, it occurs when an individual leave a community to start a new settlement. So this resultant population with time and because of its severing connection with the original population will have high frequency. Now let's have a look at what these terms are. So genetic drift, it is, as the name suggests, it's a drift of genes. So in a large population, the number of children produced by individuals with different genotypes, assuming no alteration in frequency for any particular genotype, will tend to balance out so that the gene frequency will remain stable. Large population की बात हो रही है that if population size is large, then जितने भी different genotypes हैं, जितनी भी variety है, ultimately it will become stable as natural selection will act on it to bring in uniformity. However, a small population, it is possible that by random statistical fluctuation, one allele could be transmitted to a high proportion of offspring by chance, resulting in marked change in allele frequency from one generation to next, so that Hardy-Weinberg equilibrium is disturbed. The second one is, if the population size is a small one, then by random statistical fluctuation, which is which can be equated with mutations. So in small populations, any mutation can bring in a different allele frequency, can change the allele frequency from one generation to next, and this will make the Hardy-Weinberg equilibrium disturb. This phenomena is referred to as random genetic drift. I'll make another video on all 5 factors of diversity. Then let's finally have a look at gene flow or migration. So if new alleles are introduced into a population as a consequence of migration with subsequent intermarriage, this will lead to change in relevant allele frequencies. So this slow diffusion of allele across a racial or geographical boundary is known as gene flow. So as the name suggests, gene flow, it talks about the flow of gene across barriers. So let's have two populations, A and B. Those are those are restricted by any watershed or inaccessible mountain or forest area. But with gradual communication and transportation facilities, इनके बीच में they started getting intermarriages. और उस intermarriage की वजह से जीन जो है एक population का it got diffused into one another. So this diffusion or this migration, this is known as gene flow. So most notable example, most quoted example is the incidence of B blood group allele throughout the world. So this allele is thought to have originated in Asia and slowly spread westward as a result of admixture through invasion. Okay, so this is it. Next videos में we will talk about consanguineous and non-consanguineous mating. So this is it. Thank you very much.